So really, moving forward to an overview of the talk today, really three main chapters. An overview of the immunobiology of the JAK's humanized NSG mice. And I would then share some data, how we've leveraged the two platforms that we have for humanization. So using hematopoietic stem cells and also PBMCs. And how we've leveraged those two different humanized mice to really answer some questions in terms of preclinical efficacy of immunotherapeutics. So moving on to the first chapter, just an overview of the immunobiology of the humanized NSG mice at JAKs. And I want to take a minute just to remind the audience about the JAKs NSG variants. So the NSG really is the gold standard immunocompromised mouse. And why is that the case? As the name suggests, so the end from that NSG is the background of the mice. So it's a non-obese diabetic mouse, which has defective myeloid cells. It has no complement, and really critical for permit xenograft implantation or humanization is the fact that this non-obese diabetic mouse has a CYP alpha polymorphism, which makes it human-like. So that's important when those CYP alpha expressing cells interact with macrophages that express CD47, I'm sorry. So that is really a do not eat me signal. So this is very important in permitting humanization. The S is for the skip mutation. So these mice then lack murine or B and T cells, so there are no mature murine and B or T cells. And also very critical is that these mice have a knockout for the IL-2 receptor gamma. And so this receptor is important in cytokine signaling, IL-2, IL-4, IL-7, IL-9, and really critically IL-15, which is imperative for NK cell differentiation. So these mice really lack NK cells, again, making them a good host for humanization, also for xenograft engraftment. Now, traditionally, when you humanize this NSG mouse with hematopoietic stem cells, you get really robust T cell frequencies, T cell function. You get good B cell representation, albeit the B cells do not function as they should. I'm not going to go into that in much detail. But what happens is that we then have a limited NK and limited myeloid lineage development because we don't have the chemokines and cytokines which are required to drive NK cell and myeloid lineage development. So we've then moved ahead to actually tweak that mouse to permit the differentiation and development of NK cells and myeloid lineages. So we have the NSG SGM3, so stem cell factor, GM cells have an IL-3 expressing NSG. So those human cytokines and chemokines and permit, once you humanize with hematopoietic stem cells, you're driving towards a myeloid lineage and really making this NSG SGM3 mouse really suitable for AML engraftment. And now what about the NK lineages? So when we then also have an NSG mouse which expresses the human IL-15 gene. So that mouse, when you humanize them with hematopoietic stem cells, in addition to the functional B and T cells, you also have really physiological relevant levels of IL-15 in serum, which also then would drive those levels of IL-15 of NK cells in the periphery. And now for all these mice, traditionally, once you humanize them with hematopoietic stem cells, that really permits engraftment. You could do really nice mechanistic studies. But in some cases, you're really looking to do a rapid screen, and you want to use PBMCs. And when you humanize with PBMCs, all those mice strains, they would actually develop a GVHD within the space of 21 days averagely. So we've gone ahead and also developed an NSG mouse, where we've knocked out the murine class 1, class 2, so the KBDBIA null mouse. And now when you humanize these mice and the PBMCs from adults, you also then have really an extended GVHD window. And also one advantage is that with the PBMC humanized mice from adults, so you have T cells which are actually HLA restricted, unlike the H2 restricted cells you would get from the C34 humanized mice. So just an overview of the engraftment data with these different strains. I would just bring your attention to the boxes. So we're comparing the NSG to the SGM3 and NSG to the NSG IL-15. So I really want to just focus on this box at the bottom here. So that is a frequency of CD33 cells as a percentage of CD45, so that's a myeloid cell marker. And you see that in the NSG SGM3, you really have really high levels of myeloid cell engraftment. Likewise, for the NSG IL-15, you see that you also have really good levels of NK cells, really comparable to what you find in the human periphery, which the levels of NK cells are something around 5 to 15 as a percentage of CD45 cells. And we recently also just developed a new mouse, which is not yet available to the public. We're still trying to evaluate that mouse in-house. And I'm showing you some preliminary flow data so that's a mouse which we developed by crossing the NSG SGM3 to the NSG IL-15. So the goal is then to have a mouse where upon humanization, you would have robust T cells, B cells, myeloid cells, and NK cells. And that is really what we see in this mice. So if you're comparing on the top panels, that's the SGM3, and the bottom panels, the cross, so the SGM3 IL-15 cross, and you're looking at the reconstitution of different lineages, so the T cells in black, the B cells in red, the myeloid cells in the green triangles, and the NK cells in the blue diamonds. Just bring your attention to the NK cells in the SGM3. So those levels are really subpar. And now if you look at the levels of NK cells in the SGM IL-15 cross, you find that you have really good levels of NK cells in green, as well as myeloid cells and B cells and T cells. So we think it's really a robust system then for in terms of completing a full reconstitution when you humanize with hematopoietic stem cells. Something also very important in this mouse strain is that we find that the levels of Tregs are really physiologically relevant, which is something that we did not see in the SGM3s in those mice. I didn't show that data, but they have a really high frequency of Tregs, which is not the case with the NSG SGM3 IL-15 cross. So the levels of Tregs tend to be physiologically relevant. So we're looking here in blood and in spleen. And in this case, these mice were xenograft implanted with tumors. So if you look at a percentage of Tregs percentage of CD45 for CD4s, CD8s, and Tregs, you find that the levels are somewhere around 5. Again, really comparable to what you find in the periphery in humans. So we looked at spleen, blood, spleen, and in tumor. And also something was interesting is that we find that the levels of PD-1 on the T cells in these mice are also relatively low. So this might make it a really an attractive target if you're looking at checkpoint inhibitors. So the PD-1, as we know, is a marker of exhaustion. So with low PD-1 expression, this might not also be very suitable in terms of their response to checkpoint inhibitors. And finally, just looking now at an overview of PBMC humanized mice. So we actually compared here the PBMC humanized mice in the periphery at 5 and 10 days. We're showing just a 5-day data. And we're really comparing the double knockout, which, as I mentioned, would permit the extension of GVHD window. So when we humanize these mice with PBMCs, the thought is that because we've knocked out class 1 and class 2, you would have an extended window before GVHD sets on. And so comparing the reconstitution of those mice, the double knockouts to the NSG, so double knockouts in green and the NSG in blue. So this is at day 5 post-humanization. You find the levels are very comparable, CD45, CD3, 19, 14, and 56, and 33. What you would actually remark is that there are really low levels of myeloid cells and also NK cells. And these levels actually drop further between day 5 and 10. Now, looking at body weight, which would be an indicator of GVHD, so you find that the NSG mice, after previous humanization, by day 13, in this case, you start seeing a significant drop in body weight, whereas the double knockout humanized with PBMCs, the body weight stays relatively constant. So we have some data going all the way up to 90 days. And looking at survival, so a Kaplan-Meier curve, again, the NSG humanized with PBMCs, you start seeing survival by 14 days, the mice start dying, whereas the mice, the double knockouts with PBMC humanization, they survive just fine. Again, we have data going out to 90 days. I should also say the survival also depends on the donors. So now moving on to our chapter 2, just really I'm going to breeze through some data, how we've really leveraged the CD34 humanized NSG mice to really assess the efficacy of immunotherapeutics in the oncology space. And of course, start off with cell line-derived xenografts. So what we use in this data here, the MB231, so the triple-negative breast cancer cell line, really one of our workhorses at JAX in terms of cell line-derived xenograft models. So we're comparing here two different PD-1 checkpoint inhibitor blockades, pembro and nivolumab, MB231 bearing mice, that's just your control. And we're looking at a tumor regression. So this is a mean tumor volume, and we're looking at two different donors. So what we find is that the independent of which checkpoint inhibitor we use, the response is the same. In this case, in these two donors, we see actually a nice reduction in tumor growth once you treat these tumor-bearing mice with those checkpoint inhibitors. And what is the mechanism of that response? We think it's also it's CD8 T cell dependent. So here are some nice data. When we actually have a non-humanized mouse, we check with pembro, there's really no effect. And now, if we repeat the experiment in a humanized mouse under 3 different conditions, so that's a vehicle control, and the blue is actually a control where we've actually depleted the T cells, so it's an anti-CD8 antibody-treated mouse, and the red is the same a group treated with the isotype control. You would find that when you block CD8, you actually relieve the tumor inhibition you see in the isotype treated mice. So really going to show that the effect we see in tumor growth inhibition in these mice actually is dependent on the CD8 T cells. Just some data also around the NSG STM3 mice. We have a melanoma PDX in this case. So again, when we treat those mice, the melanoma-bearing mice. Now this is the patient-derived xenograft model. We treat that with Keytruda. We see again a nice tumor growth inhibition, and really the beauty of those mice is that it actually permits you to do mechanistic studies to actually elucidate the underlying mechanism. So you could look at tumor infiltrating leukocytes. You could look at activation of your T cells in the periphery and your tumors. So in this case, we actually characterize the TILs in the tumors. So we're looking at CD33, CD3, and CD14, and you find that in this case, when you treat with Keytruda, you see an increase in the myeloid lineages in the tumors. One thing that we noticed, and as we tend to say, we learned this the hard way, is that we thought the response to checkpoint inhibitors in these mice would be uniform across donors, and really what we find is that is not the case. So I showed some previous data with 2 different donors where we saw a nice tumor growth inhibition upon treatment with 2 different checkpoint inhibitors. But what we found is that the response to the checkpoint inhibitors is really donor dependent. So we're looking here at that's a non-small cell lung carcinoma PDX model in the NSG mouse humanized with hematopoietic stem cells, and you treat with pembro, so that's donor C and donor D. You see, start with donor D, you see a nice reduction in tumor growth, which you don't see actually in donor C. So when we went back and compared the data using the different donors, what we find is that around 1 in 3 to 4 donors actually would respond to checkpoint inhibitors. And so numbers-wise, this reflects what we find in the clinic, where, as we all know, checkpoint inhibitor doesn't it's not efficacious in every patient. I think the ratio is like, response is like 1 in 3 patients would respond to pembro. So ratio-wise, the numbers are comparable in the humanized mice, but we think the mechanisms of resistance would be different, and hopefully this would present a case for some investigators to actually try to elucidate those mechanisms of resistance. So now, what about other immunotherapeutics? What about bispecific antibodies or T-cell engagers? And as I mentioned before, the MDA M231 is really our workhorse. And we did see what really was an interesting study. So we kind of termed them prophylactic and therapeutic trials. And the difference is just that for the prophylactic trials, we started dosing with the checkpoint, with the EGFR-CD3 BiTE when the tumors were really at a volume of 100 millimeters cubed. Whereas for the therapeutic trial, we let the tumors get really, really bigger before we initiated treatment. And something that we rarely find with checkpoint inhibitors, so the bispecific antibody in this case really essentially regressed tumor growth in these mice. So you see a significantly reduction of tumor growth, and independent of the tumor volume upon initiation. So even for the small tumor volumes or for the large tumor volume, you see a really nice reduction in tumor growth. Again, tumor growth inhibition, as we see here with this T-cell engager, is something that we rarely see with checkpoint inhibitors. Again, just really going to show that these mice can really be leveraged to assess the efficacy of diverse immunotherapeutic interventions. And likewise, in this case, one could also then do some flow data to really elucidate underlying mechanisms. So in the prophylactic and the therapeutic trials, we see a decrease in cell numbers. So we, of course, we see a decrease in human CD45. We see a decrease in CD3 in the human, and also in CD4s and CD8s. And accordingly, we see an activation in the T cells and also in the CD8, CD4 T cells and CD8 T cells. And those actually, the activation of the T cells and also the reduction in cell numbers was comparable for the prophylactic trials, so the top panels, and the therapeutic trials in the bottom panel. And what about CAR Ts? I mean, adoptive cell therapy is really a hot space right now. So these are published data, and this group it was published in Nature Medicine 3 years ago, and they really had an interesting approach. So they had humanized NSG SGM3 mice. They then purified the T cells, transit with their CARs, and then injected those CARs back in the tumor-bearing mice. Really, so we're looking here at some kind of an autologous system, and they had 2 different CARs. So CARs, again, are CD19, so you're targeting your B cells, and CD44, so that's a myeloid marker. And looking at the efficacy, so the graph in B here, we're looking at B cells in green. So in blue, sorry, you see a nice reduction in the B cells when you treat them with the CD19 CARs. Whereas those mice treated with the myeloid CARs, the numbers stay relatively constant. And on the flip side, when you look at the mice treated with the myeloid CARs, you see a nice reduction in myeloid cells, whereas the B cells numbers they stay relatively constant. So in-house at Wales Jazz, we've also done a lot of experiments with CAR Ts. I think I'll show some data in the next few slides. And finally, just some data to wrap up this section around the NK cells in the NSG IL-15. Again, as a reminder, so in this strain, once you humanize, it permits a differentiation of NK cells. We have a lot of data. We did phenotype these NK cells. They express the KIR ligands and the KIRs as you would find in humans. And here we're just showing a snapshot of the functionality of those NK cells in vitro and in vivo. So in vitro, that's your traditional chromium release assay at different E to T ratios. And we're comparing purified NK cells from the NSG mice in green, the NSG-15 in red, and human PBL in black. So at the different E to T ratios, the efficacy is very, very comparable. The only difference is that the NK cells, the frequency of NK cells in the NSG mice is relatively low. So actually, you would need to, sacrifice more mice. In this case, we sacrificed many more mice to actually obtain the same numbers as we compared to the NSG-15. And in vivo as well, in this case, we used DODI cells, which are NK resistant, and we treated those mice with rituximab. Which one of the mechanisms of function of rituximab is thought to be ADCC. So the thought is that in these mice that express functional NK cells, you will see actually ADCC-mediated tumor growth inhibition, which is actually what we see as well. So really going to show this the NK cells in these mice, they do function in vitro and in vivo, which ties in with data we found in terms of their phenotype. Now, the last few slides are around the PBMC humanized mice. Really, we've also done a lot of data to characterize those mice. We've shown that they permit a growth of a variety of cell-derived xenografts. We're showing here 4 different models. And there's a publication where they actually characterized 13 different CDX models. So those models grow really well in PBMC humanized mice. We could also do checkpoint inhibitor efficacy studies in those mice. So we use Jekyll cell line, the MDA, the H29, and the RKO. Likewise, even though, as I mentioned, the PBMC humanized mice are really used for those rapid experiments, you can also do some mechanistic studies. So this is an experiment where we actually compared a checkpoint inhibitor, PD1 blockade against CTLA blockade. And what we find is that I didn't show the tumor growth inhibition data, but we did some flow data to actually look at the mechanisms of action, and you could also compare what we find in this data is that it's really ties in with what you find in the tumor growth inhibition, that we see efficacy only with the avelumab-treated mice, and likewise, and we see that only in those mice you see an increase in the CD45. So I'm not going to go into much detail around this flow data. What I just want to point out is that even in the PBMC humanized mice, you can also do flow experiments to actually generate data which would tie in to the data you would find with tumor growth inhibition. And the final data slide, again, we're using our MGM231 cell line, and we're comparing the BiTE I showed previously with rituximab. And in this case, we're using the double knockout PBMC humanized mice. And as I mentioned, that BiTE really has a very high efficacy, which we see here again in PBMC humanized mice, comparable to what we find in CD34 humanized mice. So we see a nice reduction in tumor growth, which not really is the case with cetuximab, shown here in the red triangles. And body weight stays relatively constant for all the different groups, again, which is really the huge benefit of the double knockout mice, showing that there is really lack of GVHD in these mice. And again, one could also do flow data to actually tie in those tumor growth inhibition finding. And I'm just going to bring your attention here to the reduction in CD8 T cells in the BiTE treated group. And likewise, we see activation of CD4s and also CD8s. So just wrapping up, I know it's been a lot of data and relatively a wide overview across the humanized platforms. But really, in summary, with 3 main points, JAK offers a variety of NSG variants, which upon humanization with hematopoietic stem cells permit the reconstitution of different immune cells, so NK cells, B cells, T cells, myeloid cells really depends on your cell of interest. And the data really would indicate that these platforms actually present a really neat preclinical platform to assess the efficacy of different immunotherapeutics, from checkpoint inhibitors to T-cell and K-cell engagers, to ADCC-medicine antibodies and CAR-Ts and CAR-NKs. And also with the double knockout, actually, platform really permits a rapid screen of, yet really making them relevant in terms of translational relevancy for also a variety of immunotherapeutic compounds. And the beauty of that mouse is that it really, you could do those studies without GVHD compounding your data. So I will end there for today, and happy to take any questions that you might have in the audience. Thank you.